Thanks so much for that. It's a pleasure to be here. Really a lot of fun. So, let me just begin, actually. So Joyla's asked for some help with her study. What could be better than being asked to exercise? You know, I'm going to tell you today a story about developing drugs and strategies to make things better in the injured brain and spinal cord. But drugs always come with, possible side effects and things you don't necessarily want. The only really side effects that I can think of from exercise are all good. So I really encourage you to speak to Joyla. It sounds like a win-win. All right, so my lab and myself, we've been interested for many years studying the brain and the spinal cord. And it's really it's so much fun because your brain really is amazing. So, you know, when you're in the pantheon of organs, and if you're the heart, you're pretty important, you pump all the time, but the heart's a pump and we kind of understand it. The liver's a detox factory, and we kind of understand how detoxification works. And you can understand kidneys are filters and maybe a sanitation factory for your body. But the brain thinks. What's better than that? Right? And how does it do it? It's a physical thing that absolutely somehow produces the ability to make something as ethereal as a thought. So my interest has always been not necessarily how does it work, because that's a very big question, but I'm interested in how to make it better when it's not doing so good. Okay? So for a second, I want you just to appreciate, and I'm showing you this here, hopefully you can see it nicely, and you're not neuroradiologists, and neither am I, but I just want you to appreciate some things about the brain here. It's got all these kind of folds in it, and the right and the left sides look kind of like mirror images. Okay, and the next slide, so I've given you this a moment to absorb it. The next slide, there we are. Have you heard me up till now? Okay, so so the next slide is going to show you a little bit of pathology. And with this amount of MRI experience, I think almost everybody, wow, now we're getting feedback. Almost everybody's going to be able to tell me what the pathology is. Okay, so you don't even have to do multiple choice. It's pretty obvious, right? And I'll tell you that this is not an injury. Okay? This is actually a surgery. And how do you think this person's doing? So just to state the obvious, they're missing basically half the brain, right? This is not photoshopped. There is missing stuff here, right? So you would expect a large amount of problems. So I got this from Lancet. When I first saw this, by the way, I thought that it was photoshopped. I couldn't believe the picture I was looking at. And so look, so this 7-year-old, so she had a hemispherectomy. They removed one of the sides, okay, because she had epilepsy that wasn't responding to treatment. Okay, so the final path, the only final treatment they could come up with is a surgery. But look how she's doing. So the dominant hemisphere was removed with the language center and motor control for the left side of the body, but she's fully bilingual in Turkish and Dutch. And her hemiplegia, which she had at the beginning, has recovered, and she just has a little bit of spasticity in her left arm and leg. Okay, so it's an amazing thing. So how does that happen? You know, missing half the brain. This half was doing things. It's doing things in my brain right now. It's doing things in your brain. Both sides are active. She's missing half her brain, and she's bilingual. I can barely say bonjour. And I got two halves that are there. The only explanation is that somehow this half is making up for it. So in a nutshell, what I want to talk to you about is the brain's capacity to do this, the spinal cord's capacity to do this, and why doesn't it do it in all cases? Why doesn't the spinal cord injured person get up and walk after a little while? Why does the stroke victim, patient, why do they get a little bit better immediately after injury, but then it kind of falls off? You know, that is, there's some improvement and then it plateaus. Why doesn't it get back up? And can we think of ways to improve it? So let's talk about, I began, brains are really complicated. And so I began in the spinal cord. The spinal cord is, for all intents and purposes, it's part of the brain. It's really not separate. We think of it separately. It's just a little simpler, so I can sort of wrap my brain around the spinal cord, so to speak, and understand it a little bit better. So spinal cord injury, this is the point of injury in a rat spinal cord. And you can see here in green, there's been a label, so we can see the nerves that used to come from the brain up here and used to attach down here, and the injury has disrupted that. So one of the stories in this whole field of regeneration is, you know, why don't these things regrow? It's not because they can't. And that was the initial feeling. In fact, when once upon a time I went to med school, they're still waiting for me to come back, by the way. But I was taught, and we were all taught, that nerves damaged in the periphery, in your fingers, your hands, your toes, they can regrow. But in the brain and the spinal cord, they can't. It's not possible. That's what we were taught. This is now probably 35 years ago or so. We know that's not true. It's not that they can't grow. They're inhibited from growing. The body actually stops nerve growth. And one of the reasons for this is shown here in blue. So we've taken the spinal cord and we've stained it for a certain type of protein called a CSPG. I'm sorry, it's a big long word, chondroitin sulfate proteoglycan. Just think of it as the gluey material here. It's a certain protein. You find them in your scars, those kind of proteins. You find them in your cartilage, right? They're kind of a hard matrixy protein. And no kidding, when there's been an injury, the body upregulates the expression of these gluey proteins, and they actually inhibit nerve growth. So to telescope years of science, that is not mine, one of the reasons why these nerves don't regrow and connect down here is because they're stopped, because there's all these scar proteins produced. Okay? So the fun thing about science is when you learn something now, you can make a prediction. So what would you want to get rid of to make spinal cord injury better? You want to get rid of those proteins. How can we get rid of those proteins? And there are people that work on that. And so here's a publication, chondroitinase. It digests chondroitin sulfate proteoglycan. So this is an enzyme that is like Pac-Man and eats up that gluey blue protein. And if you do that in animal models, things are better. You get sprouting. Here's one for locomotor, so you walk better, better bladder. This is all done in what we call animal studies. So it does seem to work. We've taken a different approach addressing this problem. And one of the reasons for that is that chondroitinase is a big enzyme, and it's hard to figure out how it would ever be clinically applied. But it's a good proof of principle that you want to get rid of those proteins. So let me just tell you, this is a horrible biochemistry slide, the kind that we made Joyla memorize, but we won't today. We're just going to say this is what those blue proteins look like. Okay? They have a core, and they have these side chains coming off them. And they inhibit nerve growth. So we asked the question, it needs a big, long biochemical pathway to make these legs, the side chains. And so I asked the question, after injury, why does the body respond by making those proteins? It's counterintuitive. It doesn't make sense. Why do we do that? And you should know that what controls these sorts of things are your genes, right? These proteins, these gluey blue proteins that gum up the works, are merely the products of genes. So something tells those genes to come on. make yourself. So we want to know why. Who does that, right? It's kind of like you work in a kitchen. You have all these different sous chefs, okay? They're the individual genes for the individual proteins that make up that blue gluey protein. It's a large number of different genes. But you know, there's one head chef that says, hey guys, go make the souffle. And this one does part, and this one does part, and this one does part, and this. Everyone does a little part, but what you really want to know is who's the grand bonnet? Who's in charge? Who's saying go, do it? Because if you could silence him and say, no, we're not going to make a souffle tonight, we're not going to make CSPGs, if you could stop him, then you'd stop all the sous chefs. And maybe things would be better. So we took an approach of saying, who controls this process? Who makes it come on? Because if you could stop him and say, shh, no CSPGs today. Let's hold off on that. Let's wait for a little regeneration, and then you can go do your thing. So we did a whole bunch of sciencey things. I'm not going to tell you exactly what we did, although I'll tell you it's interesting technologically. It wasn't done at the bench. It wasn't what we call a wet lab experiment. It was a computer experiment. That's kind of neat. We just took the DNA sequences of all these different genes, and we analyzed them. And we said, if they're controlled together, they must have some features in common. Okay? And we actually looked in different species, because we figure whatever regulates it in the mouse and in the rat and in the human, if it regulates it, if they have the same elements in all three, it's more likely real. To make a long story short, we identified one of those, what we call switch genes, or the head chef that says, make those proteins. And I'll just give you a little bit of science. So, oops, go back. So what we can do is we can do tissue culture, where we take cells from the nervous system and grow them on plastic. And then we can put in this chef, which is called SOX9. Sorry for the name. Scientists are not that imaginative. SOX, X-O-X. And it's number 9, because you can be sure there's an 8 and a 10. So SOX9, we think, is this protein that tells all the other genes that make this blue gluey protein, hey, you've been injured, come do your thing. And so when we take cells in culture from the nervous system and we drive SOX9 expression up, so here's the control, cells that don't have a lot of SOX9, and these cells, we drive SOX9, well, we see all these other proteins that are involved in that glue come on. So that's kind of what you'd expect, right? This is the real science. If you think he's the head chef, force his activity in your cells and see if the other genes go up. And they do. What about if you block him, right? Let's try that. Yeah. What if you block him? So if we block the SOX9, all those other genes go down. Okay? So the idea then is, I've told you that one of the problems of spinal cord injury regeneration is not that the nerves can't grow, but they're actively inhibited from growing because of this gluey protein, these CSPGs. So we set off, and we looked for who controls those guys. Let's identify him. And we think we identified one called SOX9. So one of the first things you'd like to know is it expressed in this injured spinal cord? Okay, I showed you in tissue culture, it seems to regulate those genes. But is it even in the spinal cord that's injured? And so here we're looking, and here's an injured spinal cord, rat injured spinal cord. There's this big gap here because what happens after there's a spinal cord injury is the tissue gets, develops cavities because of the injury. Happens in humans, happens in rodents as well. That's why it looks very dark here. And look at all the SOX9, the red staining. And in fact, I don't know if I have a high power. No, I don't have a high power. So if you actually look, you'll see here that the SOX9, this is just a overlap of all these, so you can see one on top of the other. You can see that the SOX9 is in these little dots or circles. That's because they're in the nucleus of the cells where the genes are turned on. That's where the DNA is. So this was the first evidence that SOX9 could be actually now really involved in turning on these bad proteins in the injured spinal cord. Now, one of the reasons why we centered on SOX9, I told you that these bad proteins, we shouldn't really call them bad. That's kind of pejorative. Because they're very good in cartilage. You don't have these genes in your cartilage, then you get things like arthritis. So they're only bad in our context because they stop regeneration. So one of the reasons why we centered and we focused on SOX9 is because there's a long history of SOX9 in cartilage. That SOX9 turns on these genes in cartilage and is very necessary for cartilage. So, up to this point, if you're with me on this train, you should be thinking the following. Wow, SOX9 turns on these proteins that gum up the work and stop nerves from regenerating after injury. Wouldn't it be great if Dr. Brown could stop SOX9 in an injured spinal cord? Because then the gluey proteins wouldn't get made, and you'd get regeneration. See, science isn't really difficult. It really isn't. That's the thought. If you're on the train, that's what you're thinking. So we went to the mouse, and the first way you test this, because obviously you can't go to Shoppers Drug Mart and ask for SOX9 be gone. They don't have it. No socks away. So what we do is we go to mice, and we can, and it's a fascinating thing, you can actually make designer mice. Mice that don't have a gene of interest. It's a fantastic tool. If you wake up in the morning and you've never seen a car before, and you look out and you see a car right in front of your house, and you don't know how it works. So you look at it. So the first thing you might do is take out the steering wheel and now see what happens with the car. Or open up the hood and take out the carburetor. Do they still have carburetors? No. I'm old. So there's no, but you just start taking things out and you decide what functionality is lost. We can do that in mice with genes. We can take out a gene and now see what happens to the mouse. Okay? So what we do in this experiment is we are going to take out SOX9 in the mouse after a spinal cord injury. It's a genetic trick. It's not a drug. And the first thing you're going to ask me is, well, if you do that, do the gluey proteins go away? So here, this is kind of a boring bar graph, but all of the knockouts, there's the mice that don't have SOX9, and I only knock it down about 70%. You don't totally take it away for technical reasons. But you can see all these other proteins, and all of these proteins, these are enzymes that put on those side chains that I showed you on the centipede of the CSPG. These are sort of the core proteins. All of these different proteins, I told you there were a bunch that were involved in making that glue. All of them go down when I block SOX9. So how do the mice do? You gave them a spinal cord injury, take away SOX9, the gluey proteins go down. Your hypothesis or prediction should be they should do better. Okay. So here we are. We're looking at how they walk. We judge how they walk in a certain way. We're able to put it on a scale. This is just part of the tricks of the trade. And here are the control animals. So notice, and we talked about things get a little bit better all by themselves, and they do, even in people. So these guys are the normal mice. And these are the SOX9 mice, the mice that don't have SOX9. And they continue to do better. That's how they're walking. We're judging how they're walking at this point. So that's really fantastic. And even if we just look at overall mobility, this is looking at what we call their coordination and their ability to move their back limbs. Their back limbs are paralyzed. Their forelimbs are fine. So we look at the coordination in the hind limb. Well, just how much do they move in total? So this is totally unbiased. No one is looking at the animal. It just put them in a box, and the number of times it moves, it breaks these infrared beams. And so the computer keeps track of their distance traveled. And you can see here, this is a spinal cord injured mouse, doesn't move a whole lot. This is an uninjured mouse. And this is the spinal cord injured SOX9 knockout, the guy that doesn't have SOX9. So it's as mobile. It's fascinating. So, but science never misses an opportunity to surprise you. So if you're sitting there and you are still on my train, we said spinal cord injury, these guys do not regenerate through the lesion because there's these bluey, gluey proteins, the CSPGs. SOX9 regulates these. So when Dr. Brown got rid of the SOX9, the bluey proteins went away, the mice walked better. It must be because these guys grew through the lesion because there was no scar tissue there. And that's what we thought. That's what we expected. It's not what happened. Okay? And I think sometimes it's good to ask yourself, what's a reasonable ask? What do you want to achieve if you're going to try to treat something like spinal cord injury? Is it reasonable to think that this nerve here, once I get rid of the blue proteins, is going to grow through the lesion? How is it going to actually jump across? I don't know. But it grow all the way down and find its original target? There's lots of nerve cells in that spinal cord. Even though I said it's simple, it's not. So I don't even know if that's a reasonable thing. But we took a look and we said, well, let's see how many nerves are growing through that lesion in the SOX9 knockouts where they walk better. This is a little bit sciencey, but let's just look at it for a second. The green stain, where we've cut the spinal cord in across like this. So the nerves growing through are going to look like dots. Okay? And here is above the lesion. And you can see there's a lot more green in the knockouts than in the normals, the ones that have normal SOX9. Ooh, there's more green in the SOX9 knockouts. And below the lesion, there's more green. But at the lesion, growing through it, there isn't. Okay? Here it is here. At 0 point, this is ground zero here, right at the lesion, it's about the same. So there's more above and more below, but there's nothing more going through. That was surprising. They're walking better. Obviously, there's more connections from the brain that now can grow through. That's not what's happening. There's not an increase growing through the lesion. Why are they better? That was the problem a few years ago. This, by the way, is a story that's developed over about 10 years. So everything was going along fine. Knockout SOX9, no blue proteins, mice walk better, must be regeneration, and I don't see it. So now I'm stuck with a great result, but no explanation. So then you have this sort of uncomfortable truth. I told you that CSPGs are in that scar where the lesion is, those blue proteins. The truth is, that protein is part of the normal nervous system. Yes, there's lots of it turned on at the scar when there's an injury. But normally, in the normal nervous system, your nerve cells are like enmeshed in these CSPGs. So here it's shown in red. I should probably change the color to blue so I can tell you. But just a different color. Notice this red-like mesh around this nerve cell. All of these are CSPGs. They look like they got little holes. Your nerve cells are surrounded by material that inhibits nerve growth. That's surprising again. It doesn't make sense. How can you have your nerve cells surrounded in this glue that doesn't let nerve cells grow? How does that happen? It doesn't make sense until you think about what happens in the embryo. In the embryo, so here's my little diagram of an embryo, or at least the nervous system in an embryo. And here's a nerve cell here. And he's going to get connected. So when you're an embryo, and you all were, the nervous system wired, right? In utero. Your whole nervous system, your nerve cells are born, and the most important thing is how they connect up. And they do this in the embryo. The embryo is really smart. It knows how to do this. And the reason why it's able to wire itself is because in the embryo, there are no CSPGs. So in the absence of CSPGs, these guys can grow out, and they can find their targets just fine. And once they find their targets, you need to stop them from finding targets, right? Once the nervous system wires, you want it to stop wiring. You don't want excessive growth. You want proper. So once the nervous system wires, the next step is it makes its bluey proteins around all its nerve cells. So it says, hey, we're wired, we're good. Now we're going to stabilize our connections and make sure we don't have any aberrant connections. Great. So now you have nice red CSPGs, that net, and the reason why the net looks like it has holes is because those are the connections. So CSPGs are part of the normal program of the nervous system. It helps it mature. But what happens after an injury? Right? If you have an injury, now these guys have gotten disconnected. And nothing new can grow in because the CSPGs are there. They're like a holdover from the embryonic days. So we come and we say, and when you have an injury, by the way, you also get CSPGs at the lesion site. So now let's think about when you stop SOX9. Why does it work? Well, when you stop SOX9, this goes less. When this goes less, now this guy can sprout. Because the nerves like to grow. They're stopped because normally there's CSPGs, there's that glue. If you decrease the glue, now they can sprout. And you get something like this. Boom. So what happens in the knockouts is not that they get growth through here, that long-range hypothetical, what I was dreaming back 10 years ago might be able to achieve, but what you're going to be able to achieve maybe is that things that are spared the injury are going to be able to sprout because there's less inhibitory. That's an easier ask too. Because if you're going to think that things have to grow a long distance and find their old targets, good luck. But if you want to think that whatever spared the injury might be able to sprout, that's possible. That's a more achievable goal, in my opinion. And so one way we can look at this, and let's move on. This is the science. I'll talk just very quickly. This will be for Joyla. So here we go. So we're going to trace these axons. So what I want to do is I'm going to put a little dye in the mousy brain, and I'm going to track it down below the spinal cord injury. If it's because of sprouting that this guy walks better, then there'll be a lot more terminations, a lot of sprouting below the lesion compared to normal. And here you go. You can see these terminations as red dots here. I don't know if we can turn down the lights. Oh, it probably doesn't matter. You can see these individual red dots, and in the knockouts, you can see that it's about 3 times as many for the same amount of dye. So things are sprouting below the lesion, not growing through new. That you have to get rid of that idea for now. Maybe one day we'll figure that out. But it's sprouting of what's spared. And the good news to me about that is it means that the ask is smaller. Whatever's targeted correctly, just increase. Okay, so let's just now briefly, what about other forms? So the nice thing about working on this sort of basic science level is, these principles of nerve growth and CSPGs, they are basic science. They exist regardless of the form of injury we're looking at recovery from. So what about stroke? So we looked at it in stroke. And again, we could take now we've made the mice, so the mice are easy. The knockouts, they don't have SOX9. We can give them a stroke-like injury. How do they recover? Well, here is something we call a corner test. It doesn't really matter. These are all tests of whether or not they can use their right and their left sides equally after a left-sided injury. And in fact, you can see that the knockouts here are more like a 50-50, 5 out of 10, use of right and left sides, whereas the normal guys, the ones that have normal SOX9, they are still favoring one side over the other. And we can show that in multiple tests. There is always this, the knockouts, when you don't have SOX9, they have more of a symmetrical use of limbs than the ones that have normal SOX9. And even here's something easy to understand, grip strength. Right? So they've had a left-sided stroke, so we know their right paw is going to be weak, okay, because it's always the opposite side, even in humans. And in fact, you'll see that the knockouts recover strength so much quicker than the normals. So even in stroke, and let me show you what we've shown in stroke, it really is fascinating. We do an injury on the left side, and I'm going to show you two types of tracts. There's a tract here that projects from the motor area of the brain to another motor area of the brain. This is just called the red nucleus. Doesn't really matter. This is same-sided. And when you've done a left-sided stroke, this one has lost its inputs. These guys aren't there. The blue and the greens aren't on this side because they've been damaged by the stroke. And what happens is they sprout over. They actually sprout onto the opposite side. So if you take a look at it, this normally goes from same side to the red nucleus. And when you decrease SOX9, what happens is it's going to sprout over. And you're going to be able to see that. And in the spinal cord, so you're probably more familiar with this idea, this left side would normally project to this side to control the opposite side of the body for movement. So the right paw would be controlled by the left, but he's been injured. And what do we find happens? This side that normally goes to the opposite side now sprouts onto the other side when you knock out the SOX9, because you've removed the CSPGs from around those nerves, and now there's the opportunity for sprouting. So the brain can actually rewire itself. Because what's happened, in short, just step through that, doesn't matter. What's happened is in the SOX9s, there's been a decrease in the normally occurring CSPGs. And the other thing is that they've lost the normal inputs because of the stroke. And so these guys that are from the uninjured side of the brain, they sprout to make up, to compensate. If that's not amazing, I don't know what is. Let's skip through. 10 minutes. Okay. That's all in mice. We don't really want to cure spinal cord injury in mice. Just keep them out of my lab, and that'll do it. Does any of this exist in humans? Okay. So we do have some pathology samples. This is an uninjured human brain. We're looking for SOX9. There is not a lot. GFAP is a marker for support cells, not nerve cells, but support cells that are in the brain and spinal cord. Here's an injured spinal cord. Spinal cord injury. This person suffered a spinal cord injury about 20 years prior to their death for unrelated reasons. We got a little section of the spinal cord, and you can see in red all the SOX9. That's a stain for SOX9. This is a sample of someone who suffered a traumatic brain injury. The red is SOX9. This is someone who had a stroke, two different types of stroke. Again, the red shows you the SOX9. And this is actually a sample from an Alzheimer's brain. This is one of the famous kind of plaques. And again, you can see lots of SOX9 in those astrocytes. So, mice and humans are quite a distance apart, but many of these genetic programs are the same. So why is it on in so many different things? I told you about spinal cord injury. I've told you about stroke. I've shown you some stuff in traumatic brain injury. I could show you the same story in concussion. It's because these are basic pathways that are conserved. It's probably inflammation that turns on the expression of SOX9. And SOX9 then turns on the expression of CSPGs. If we're able to block the expression of SOX9, we can stop some of that and get what we call reparative sprouting of nerves and make things a little better. How do you then move this forward? We cannot do these genetic tricks in humans. You can't make human knockouts for SOX9. So what you have to do is you have to start to think about ways that you could pharmacologically block SOX9. Okay? And so we have, over the last 4 or 5 years, been trying to develop pharmaceuticals that would block SOX9. Again, the dream is, if you can block SOX9, you block the expression of those proteins, you can get sprouting after things like stroke and spinal cord injury, or maybe even brain injury, and make things better. It's a tricky process. I forget exactly, yeah. So, what we did, I don't know, let me just see for a second. Yeah, so where we went with this, one sec, let me just see. Yeah, I don't have that slide. Okay, so where we went with this is we did, again, and it's, computers have really revolutionized how we do science. And this is, again, all completely a computer experiment. We took SOX9's structure and we did a computer simulated test of about 12.5 million compounds, trying to let the computer guess for us which compounds would bind to this SOX9. So it's crazy. You don't have to do any bench work for some of these things. And you just do, you have a computer program. This is done with actually an Ontario biotech company. We gave them the structure of SOX9. We told them what part of SOX9 we thought would be important. And then they did some computer program, and they identified a particular compound, or actually 10 of them, with high probability, the computer said, of hitting SOX9. And so then what we do is we come and we treat it like Tinker Toys, where we take off this sulfur and we put an oxygen, and we take off this methoxy group, and we start making different types of molecules, and then we can test them in our models. So what I want to end with, and we can try to go to humans one day. So we found one, and several, but we'll talk about this one called Z02. And we go back now to the very first experiment I told you about for SOX9, where we take cells growing in plastic. Now we treat them with a drug, Z02, and we say, well, what happens to the expression of those gluey genes? Woo, they go down. Okay. I know my train's getting long, but we're getting close to the caboose. So about 2 cars away. So if you're still on the train, right, we identified this guy SOX9 that turns on those glue genes. When we genetically got rid of that SOX9, the gluey genes went away. The animal walked better, stroke was better. And now I'm showing you that we can find a pharmaceutical that we can now use to stop the expression of that genes in cells in plastic. Okay, but that's plastic, right? So this was really exciting. I gotta say, this is brand new. You're seeing this. This is the first time I'm showing this slide. Actually, it's possibly in the hands of some of the viewers right now. We are hopeful. This is the spinal cord of half of a spinal cord. This is where that's actually a motor neuron there in an animal that had a spinal cord injury. And then we took these animals with spinal cord injury and we treated them with the Z02. And we stained the section for these CSPGs. Now, they're normally there, remember, as part of the normal development. But we treated it like for 3 or 4 weeks. And you can see, here's what it normally looks like. The nice green, gluey protein now keeps changing color. It began blue, then it's red, now it's green. I'm really trying to confuse you. So it looks green right now. And you can see how it really is around the nerves mostly, right? You see how it makes this kind of net around the neurons. And here it is, another guy, another animal. And these are the ones that got Z02. Okay. So showed it to you in plastic. Now we're showing it to you in an actual animal. You get it Z02, you can knock down the CSPG expression. So now, oh, and this is just that in higher. I'm so happy with this result. This is actually a higher. I put this, making a wallpaper of this for my office as well. Again, untreated animals, you have lots of this gluey CSPGs around the nerve cells, and these guys are not doing so well. They don't walk as well. And these are going to be the ones that are treated with Z02. And you can see there's a lot less green. Not totally gone. You can still see some in the background, but it's like 3 or 4 times less. Do they walk better? So this is our first experiment. So I wouldn't stand on this. Small groups of about 8 animals or so. But we are beginning to see that the groups are separating, where the Z02 animals are just beginning to walk a little bit better than the ones that don't get Z02. Okay. So hopeful. So what have I showed you? And you know, really, we go back to the first slide. The brain has an incredible ability to actually repair itself, which is a good thing. There's like 10 to the 11th different nerve cells in your brain. It's a huge number. That's 100,000 million cells. And they're all interconnected in very complicated ways. If you had to sit there and connect one to another, you'd never do it. So the fact that there's wisdom in the brain, that it knows how to do this, all we got to do is just maybe remove a little bit of the inhibitory factors that are holdovers in some sense from an embryological time, a time when you needed the growth and you needed the connection. So if we can kind of turn back the clock by getting rid of the CSPGs to sort of mimic this embryological state, maybe the brain will rewire itself, and that's partly what our animal studies are showing us. So it's kind of like this. The final, in case you fell off the first train, first car, and you're waking up again. The story is that the brain has an incredible ability to wire itself. And it's kind of like you have a house, and the house is nicely wired. You get lights in the attic, lights for the bedrooms. You have electricity for your front door, and everything's great. And then someone comes along, and they cut some of the wires. So now you're in the dark. You're in the dining room, and you can't see. Wires have been broken. Well, what the brain can do is, can you imagine, right, that a wire up here senses that this guy's missing inputs. And it basically glows down, and it reconnects. Right? Your house doesn't do that. So it's an incredible power that we have, and what we're really trying to do is get at ways to maximize that and open up that window. So just for a second, I think I got exactly a minute and a half. Wow, look at me. So you can see we've been doing this for a while. These are my colleagues. Actually, I got into spinal cord injury because of Lynn Weaver. I was basically an embryology guy. I was studying how things connect up in embryos. And I came to Western, and Lynn was a great colleague, and she showed me the ropes for spinal cord injury. Greg Deckerman is an immunologist, and Paula Foster is an imager. And it's really nice. We have a team that works on spinal cord injury, brain injury, concussion. And it's such a great way to do science in a group as opposed to being off by yourself. And I never would have done any of this without these people. And even more importantly are the actual people that do the work. So here, let me just point out, where's Todd? So Todd has done all the work on Z02 and spinal cord injury. Nicole does a lot of our work on the histology and the staining for all those things that you've seen. And then Cathy Zhu did this stroke modeling that I showed you. And you know, the truth is, so I sit in the office and write grants and drink coffee and eat donuts, and these are the people who actually do the work and to whom I'm really indebted. Happy to take any questions. Thanks for your attention.